Log on, tune in, find out. Another good idea from Cambridge.
So it's a great pleasure to be able to talk to Sidney Brenner, who I've known for over 30 years now in King's College. Thank you, Sidney, for doing this interview. Could you tell me first where and when you were born?
I was born in Germiston, which is a small town outside Johannesburg in South Africa, and I was born on the 13th of January 1927.
And firstly, something about your parents and their background would be very interesting.
Well, and my father came out from Lithuania as an immigrant to South Africa. Went to South Africa because the fare was cheaper, and he had a brother there, and that we reconstructed to be about 1911. My mother came from Latvia, at least on the border of Latvia and Russia, and she came out in 1922, so she lived in Russia during the war, the revolution, and that is what I remember from my youth, being regaled with stories about white Latvians and red Latvians and white gods and red gods. And my father was a shoe repairer, and we lived initially in rooms at the back of his shop, so I grew up with the smell of leather, and that was my early life there.
Tell me something about their characters and how they influenced you.
Well, my mother was someone who had, I think, great ambitions for her children. My father was illiterate. He couldn't read or write, but he had a great gift with languages. He spoke several languages, including an African language. My father was he did that, but I think the important thing is that my mother encouraged me to read. She knew how to read, and I learned to read because I visited another old lady who was a friend of my family, again another immigrant from Russia, and learned to read from the newspapers that were spread out on the table in lieu of tablecloths.
And then what was your first school? Do you remember your first school?
Yeah, I went to a kindergarten because a lady who actually ran it, it was attached to the Presbyterian Church in there. Her name was Miss Walkinshaw, and she ran the kindergarten, and she came in as a customer to my father, and she found me at age 4 and a bit reading a newspaper on the floor, and so she said my father should send me to her kindergarten. And of course my father said that they couldn't afford to, but she took me in there, and I did the first 3 years of primary school, which was in South Africa was grade 1, grade 2 and standard 1. I did that all in a year, so that when I went to the government primary school, I went straight into standard 2 at the age of 6. So I managed to compress 3 years in 1 year, and so I was always about 2 years ahead, younger than everybody in the same class. which was not helpful.
Were you quite small at that time?
I was small at that time and a bit of a coward, I say, but you know, often got beaten up. It was that kind of school, yeah.
And then after that school, where did you go on to
Well,
school?
then I went to a high school called Germiston High School, and at the end of that I matriculated when I was just less than 15, but I'd won a scholarship, a bursary to the university, given by the local town council, which meant that I could go to the university in Johannesburg to study medicine. Of course, by that time had a very big interest in science, a major interest in doing that, but medicine was what was being financed. And so that's how I got into biology, if you like, although I had a lab of my own. That is, I had advanced education in garage chemistry.
Garage?
Garage chemistry.
Garage chemistry, huh?
That is, doing it in a garage, yes.
Were there any teachers at that high school who particularly influenced you?
I can't remember any of them that influenced me. In fact, I was completely astounded by their ignorance because I received most of my education from the Germiston public library, which was the endowment of Carnegie, and spent nearly all my time reading there.
You mentioned that you were already interested in science. How would that happen? Just curiosity, or?
Well, I think it is, you know, I think it is, most children are interested in nature. And of course one of my ambitions was to find out about nature. So the way to do it is, of course, to take it apart. I remember a few years ago I was in Japan and several elderly gentlemen were discussing how they became interested in science, and one of the physicists said he got his interest in science by taking a crystal set apart and putting it back together again. And I said, well, I did the same thing because I used to take flies apart, but I couldn't put them back together again. So that became one of my interests. How would you put it back together again?
So at the age of 15, 16, you were ready to go to...
When I was 15, I started.
At university?
At university.
You must have been by far the youngest.
Yes, I was 2 years younger, at least 2 years younger than most of the people there.
Which university was it?
There's one in Johannesburg, University of Witwatersrand. And I lived at home, I travelled every day, first on a bicycle about 2.5 miles to the station, then on a train, and then walking to the university from the railway station, and taking sandwiches for lunch. And going in the morning, we had a much tougher regime there. We had lectures or laboratory sessions every day and Saturday morning. Everything started at 8 o'clock there in the morning. So it was quite, one was to have to set out at about 6.
Did you enjoy it?
Yes, because I think there I actually did meet people who were doing things. There was a man in the botany department who I discovered just in walking past, and he let me come and work in his lab, who was doing chromatography, which I thought was astounding, separating all these colors from plants. So that I did enjoy. and I learned, of course, we did 4 subjects: botany, zoology, chemistry and physics. I liked the zoology, but of course we had to learn a lot of things there in order to write examinations in vast details, you know, all the embryology of things and so on. And so after the first year, I then moved to the medical school, where I did anatomy and physiology, and that was interesting as well. And then it had been discovered that I couldn't qualify because I would have been under 21 when I finished medicine. And so I was able to take a year out to do a Bachelor of Science degree in anatomy and physiology. And that became heaven, because there one worked in a lab, and I took out 3 years. I did a BSc, then a BSc Honours, and then a Master of Science. But then I think I was already doing scientific research. And I realized then that I wasn't a good medical student. In fact, the other day I gave a talk entitled "The Worst Medical Student of 1950," which, but in fact, at the end, although I resented, you know, having to go and complete it, and I should say, I completed it because I'd finished all these other science things by the time the war ended. And everything had opened up by then, and people had started to go abroad again and so on. And the question was, I thought, since many of my friends were going, what should I do when I finish my master's degree? And should I go abroad and try to do advanced work? But I was advised to never get a job as a scientist. And the only jobs for my interests, which then would be in medical schools, and you had to have a medical degree, otherwise you were a second-class citizen. So I stayed back to finish 4 more years of medicine, from 1947 to the end of 1950, and of course then I did go abroad in 1952.
So how long had you been altogether at that university?
I was at that university from 1942, yeah, I think almost 9 years. Where I'd done a medical degree. I landed up there actually teaching. I was a lecturer. In fact, I became a sort of lecturer while I was still a medical student. I used to have to go and teach people physiology. But that was good because, you know, when you teach a subject, you have to really know it and understand it. And I mean, I became a great expert on one of the things which nobody could understand, which is, you know, calorie intake and all of this. Now become very important. So I knew exactly why you burnt all the things at the rate you did and didn't at the rate you didn't.
Well, were there by this time any teachers or colleagues who you particularly remember as influencing you at university?
Yes, well, there were 2 people there. The one is, there were many people there that influenced me. The most important one, I think, in that time was Raymond Dodd, who was the professor of anatomy, but more so was a man called Joseph Gilman, who was then a lecturer in histology in the anatomy department, later became professor of physiology. And he was the one who encouraged everybody to read widely but of course didn't like to be contradicted. And so many arguments became quite heated after a while. But I think discussing scientific problems and actually working in the laboratory was a tremendous experience, and that I think, and also doing things right from the start, because there was nothing there. I wanted to do an experiment which required an amino acid, but of course no amino acids had reached South Africa since the beginning of the war. And so I prepared the amino acid. I found a book, I read it, I went to the barber shop, I collected all the hair, and I cleaned it, of course, and then hydrolyzed it and made cysteine. In fact, I got so good at that, I was making all the amino acids from various things like milk, powder, hair dropping, well, hair cuttings for nutrition experiments.
It's rather like Newton grinding his own lenses.
Yeah, well, I think this is what we did. I even built an ultracentrifuge and used it in what I considered to be one of my really clever experiments.
Were your parents happy that you were spending so much time at
I think
education?
they were very generous in the sense that, of course, in classic Jewish families, learning is highly regarded. But my mother would have been much happier to see me at that time as, let us say, a specialist doctor, because that had very high social status, you know, with a nice car and a good practice and so on. And I think that I think it was only very late in life she realized that I'd got somewhere after all.
So now you move on from there to postgraduate?
Well, yes. Well,
postdoctoral.
what happened was, I think I know what I was interested in, but the subject hadn't been invented. It's called molecular biology. Waddington, that's Caroline Humphrey's father,
Yes.
came out to South Africa at the time, and he encouraged me to go to apply to Cambridge, which I did, and of course they never even replied to my letter, so that wrote Cambridge off. But I won a very rare, there was only one per year for the whole of South Africa. It was the scholarship of the 1851 exhibition. So I won it for that year, 1950. I applied for it. And where it came to go is, where it came to for me to decide where to go, I asked the advice of the principal of my university, who was an old Oxford man. And he recommended me to go to work with Cyril Hinchelwood.
Cyril
Hinchelwood.
Hinchelwood, yes.
Hinchelwood was the professor of physical chemistry and had written a book called The Chemical Kinetics of the Bacterial Cell, which sort of fused physical chemistry and biology. And so I applied to him and got accepted. And so I then went in 1952 to Oxford to do a DPhil in physical chemistry.
Was it something of a shock to arrive at Oxford?
Well, I'll tell you, you know, we had a group of people in South Africa, because apart from doing all the science, I was with a very talented group of people, some of whom are still alive, where we actually made films. And we decided, for example, to make a film of one of, I think it is it I think it was by James Joyce, but I'll need to try and recall it. However, but it was about England and it was about Wales.
Yes.
So it wasn't about James Joyce. I think it was Dylan Thomas,
Yes.
actually.
Mm.
And so, of course, we had to imagine
Mm.
what England was like without ever having seen it.
Mm.
And however, you know, we had this, of course, everybody was connected with England by just by books and by the sort of education we'd had in the school. But it was a shock when I actually came there, because the one thing that I found almost desperate was food rationing was the law. And for 2 years I just dreamt of food, because, you know, one had very little. However, after a term in Oxford, May and I got married. She was in London doing a PhD. And so we got married and we set up a house in Oxford where we both finished our PhDs in 2 years at the time.
Gosh.
And I then won a scholarship, a traveling scholarship from the Carnegie Corporation to go to America for 4 months. In the meantime, as I've said many times, in about November 1952, I had a very good friend in Oxford called Jack Dunnett, was a crystallographer and in contact with all the major figures in the field. And he told me, I came to Oxford with two half-wrong theories. I mean, it's important to measure the amount of wrongness in a theory, because even a tiny amount really kills it. So I came to England with the idea I could determine the structure of DNA by some method, but it was all half-baked.
It was already a well-known problem.
No, it wasn't a well-known problem. It was a rather fringe problem, because, I mean, genetics had its problems, but to connect it with this chemical, because it wasn't actually accepted that the genes were built out of DNA until much further on.
Yes.
And people fought a dying battle. That's simply because in the field, most people felt genes do complicated things. They better be built out of something quite complicated, like a protein, you see. And of course, J.B.S. Haldane had coined the concept, I think, as early as 1929. It's the greatest misleading concept in all science, in which he characterized genes as autosynthetic enzymes, which is wrong. And so all of this, you know, just I said that this, but of course I didn't have that. But of course I had become interested in it. And my interest was really taken up when, of course, I went to Cambridge. After hearing about Watson and Crick, we actually went in April of 1953, just before the paper was published. 3 of us, Jack, Leslie Orgall, and myself, went to Cambridge and met Watson and Crick and the model. And of course, that was a revelation. And it was, you know, one saw the light, and since then one never turned back.
They had already discovered?
They had already discovered the structure. And we saw the structure, and its implications were just blindingly, I won't say obvious, but blindingly clear. And you sort of, after 5 minutes you sort of realize, well, there's the coding problem, there's the copying problem, so we better get down and solve all of these. And that's essentially was the real turning point in what one was going to do.
What was your first impression of those famous two people? Because you worked a lot with Crick later, of course. But
yeah, well, Francis, I mean, for that one day, of course, Francis wouldn't stop talking, basically. But Jim just always gave me the impression then, and I wrote about it later, of a kind of irritated bird. You know, pecking this, pecking that. But the impression was they'd made a breakthrough. You know, this was what the world needed, although no notice of it was taken at all.
Really?
For quite a time, yeah. It was the most amazing thing, because everybody sort of doubted, A, the way it was produced, B, there were problems there, but to me it was just very clear this is what we have to get on with. And so for quite a time, the field was essentially occupied by a tiny band of people. You could 20, 30 people who saw that this had reformulated major questions in biology. And although one always writes these things with hindsight, it became clear that the sort of problem that most people were preoccupied with in biological systems at the time were problems of matter, which is how do you make all these various, you know, compounds that you find in the body, B, where you get the energy for them. But no one had said, look, the important thing is how to make these proteins, how to get the amino acids in the order. This is the coding problem. And we knew that that was the problem, and we knew it only from the work of Sanger. So while I was at Oxford, there was a club there called the Alembic Club of Chemists, and Fred Sanger came to talk, I think it was in 1953. Yeah, that's correct. And he came to talk there because he had just assembled insulin, made the sequence of insulin. And he had wonderful gadgets for his lecture, which consisted of little blocks that as he made the pieces, he turned them around and you could read the amino acid on the little block, you see. And it was a great thing. But I'll never forget that after his lecture, Robert Robinson, who was the doyen of British chemists, stood up and said that this was remarkable, because Sanger has proven proteins actually have a chemical structure. No one believed in it. They were some kind of gloop or glob which had just polymerized together. No one had any idea that they had a defined structure in the form of a sequence. And that's the thing that matters. And once we had that, then you have to ask yourself, how do you get these things in a row at the same time? Now, I'd also had a background. I had a very good friend in South Africa, who was a mathematician, called Seymour Papert. And I got very interested in certain aspects of this. And in particular, I did read a very interesting article by a man called John van Neumann. And I still have the book, which was written before the Watson-Crick. It was how you might build an autosynthetic machine, a machine that could build itself. And of course the logic of this is exactly what was embedded in the DNA. But of course nobody knew that at the time, so I'm sure that if someone is just blatantly writing the history of ideas, he could actually look at Van Neumann's publication and say, my God, there he's written about DNA. And there's Watson and Crick influenced by Van Neumann. But in fact, the most amazing thing is that in 1949 it is, Pauling and Delbruck had written a paper about complementary replication, which is I make you, you make me, and we've got a double helix if you like. But they'd forgotten about this. It's something I always tell students, you can read about ideas, but it's how seriously the proposers take them that counts. If they are just thrown in, or we can get a machine that makes random ideas, but you can't lay claim to them unless you are a supporter of them.
You mentioned Fred Sanger, and I'm hoping to talk to him.
Yeah, good.
Can you say something about him? He always struck me, he's at King's of course, as a very modest, quiet man.
Well, he was a unique scientist, Fred, and I've always been impressed. There's no one else like him, in the sense that he saw that, and as he did first in protein chemistry and then later on in the whole of DNA chemistry, that determining the sequence, determining how the components are arranged is important. And he devised what were very simple techniques in order to achieve this, and carried them out. And he liked to work in the lab. I mean, when he retired, he put down his pipette and said, that's it, and walked out. But I think he was a real scientist, and I think the things he had had major impact. But the one thing about Fred, which is very amusing, was I noticed that he went up by factors of 3 when he started to sequence. He first did something which, let us say, was 6,000 bases long. He first did something in the RNA field, which was very small, and then he got up to 100, you see. Later on, he kept on going. He got up as far as 50,000 this way. And I had a colleague, and I had an idea about how we might purify a product of a gene. And I told this young man from America who was working with me at the time that we should go out and talk to Fred to see if he'd be interested. And I said to him, when he asks you how big is it, say it's 300. So, but he chickened out at the end because he thought this was too big. So Fred said, how big is it? How big do you think it is? And he said 100. So Fred said, oh, we're not interested. We've done 100 already. So that was
fascinating. One other question about DNA. I've heard sometimes that, and it's mentioned in the double helix, that I think he was a Trinity originally, and then later a King's mathematician, John Griffiths, who you knew.
John, yes.
He had an important role in...
Well, yes. John was... I knew John very well, and John was a very good mathematician, and he is... he's famous for 2 things. Oh, well, famous. Nobody's famous who hasn't done work over the last 2 years because everybody else there belongs to the common epoch, which is the one preceding the last 2 years. And he he solved or he helped to solve, there's a very famous paper by Crick, Griffith, Ogle. It's called the Commonwealth Code. And that was, if you took 4 things and they wanted to code for 20 amino acids, how could you arrange them so they made other conditions? And one of the conditions was that you should be able to start anywhere and it would still make sense. Now, of course, taking 3 bases for each codon gives you 64. And that's too many for the amino acids. But if you make that code commonless, which you can do very easily because it says if the sequence, just to give you one example, if the sequence consists of the same letter, the triplet, like AAA, you can't make that commonless because you wouldn't know where to start if you had 2 together. So there'd be several ways of reading this. And so that eliminates 4 out of the 64, leaving you with 60, which happen to divide into 20 classes, each containing 3, and that was the Commonwealth Code. Marvelous, except it's wrong. And that's the great thing, that you should always suspect that an elegant solution is probably not nature's solution. Because as I often say, mathematics may be the art of perfection, but biology is the art of the satisfactory.
Although elegance is a nature, your famous nematode worm was
called C. elegans.
Yes.
I didn't give it the name. It was given to it by a Frenchman who discovered it, yes.
Okay, well, let's go back to your life. You've done your PhD and you went to America.
I went to America and then I went for 4 months and did a lot of work there. And then, but in the meantime, I started to discuss with Francis about coming back to England, in fact, joining him in the MRC unit. And so I went back to South Africa because I had to obey certain of the conditions of the fellowships I had. And 2 years later, after arrangements had been made, I came to Cambridge at the end of 1956 to a 3-year job at 1,100 a year with 3 children. So, but I came to do, and I think that was the really beginning of what was an incredibly exciting time in science.
Working with Crick, you mentioned various things, for instance his famous notice on the wall, which I have on the wall in my office now, I'm reading rots the mind.
Yeah.
But you said he was actually a rather well-read.
Yes, well, this is what I've written a little piece on this, you see. I didn't know where he got this from. But he turned up one day and he put it up on the wall, reading rots the mind. I later learned that he got it from Christopher Longevitt-Higgins, who was a mathematical physicist and professor of theoretical chemistry. Because Christopher really believed there were only 2 things you could think, or you could play the piano, basically. But reading rots the mind. So Francis had this because of the things, but of course it was hilarious because he would sit underneath this reading all day. And when he went to the Salk Institute in the mid-70s, the sign was still there above his desk, but the entire room was full of books on the brain, through which he was steadily reading. So we have to
Guess what he meant.
we have to guess what that was.
But what you struck me about your account of your relationship is these conversations,
Yes.
working things out in words in conversation.
Well, I think I think that was a very unique combination, because the rule was you could say anything, I mean, provided it wasn't stupid. And in many cases, one would get it half wrong. You see, someone like John Griffiths will go away and think about it and then come and produce the finished version. I can't think that way very easily. I have to churn it around, and I churn it best within conversation. And I think we produced many things that way, and even up to almost the last week I spent. I saw Francis in there, and we spent hours discussing, the brain and how one would actually do things, and so that's, as I think, a very productive relationship, because I think we've only written about 3 papers together, although we were colleagues in the same thing, and there's only 1 paper which is just by the 2 of us. It was just a short thing, but it was pretty nice to have done it.
Did you, after you'd had these conversations, would you rush off and write ideas down from the conversations, or?
No, Francis would be the one. I would actually go and try and see if I could do an experiment to pin down those ideas. And that's been my habit ever since, which is ideas are fairly cheap, and they circulate, so to speak, in some space of their own. And the thing that really clinches them is the experiment which proves they're right. And of course, this idea that you can prove something correct by science is now sadly disappearing. Everybody thinks their task is to describe nature in ever increasing minutiae.
You say a number of times that it's very important to just have lots of guesses, many of which are wrong and some of them are half right. I was very struck, one of my favourite quotations from Einstein is, unless an idea starts off as absurd, there is no chance for it.
Yeah, correct, you see, because most ideas are wrong in the beginning. I mean, you know what you want to try and get, but in fact you've started on a slightly wrong path, and you've got to a thing and you sort of are very triumphant till you've thought about it and you discover there's a little snag in it, and so you must reiterate it. And so I think many things go, and I think a talking exchange and being able to tap evidence from all sources, which one could do with a number of the people that I'd had contact in my life, is I think very valuable, because this is an interesting way of generating a cooperation, a cooperative thing, which usually produces something that's quite interesting. I mean, and often right.
Well, you make a distinction between correct theories and true theories.
Yes, you see, the Commonwealth Code is correct, but it's not true. That it's logically correct. Many theories are logically wrong. Many can be wrong because there's some fatal flaw either, in the deductive or whatever it is. But the one that coincides with nature is what we're interested in. And we want to know how nature does it. And therefore, it's true, you could probably do it better than nature, but that's not the name of the game.
Coming back to your conversations with Crick, did you develop a sort of, not exactly secret language, but a shorthand?
Well,
Because-
after a while, you see, well, we got one of the things that, I've invented a story about Francis, which is he would sit at his desk and ponder on one of the problems, and then would say something. Now, he was once talking about the imaginal discs of flies and how they might develop, and then when he got frustrated, he quite often banged the desk and said, God knows how these imaginal discs work. So from that I created a story of Francis going to heaven, so on, and then finally meeting with God and saying, how do these imaginal discs work? But of course God then says, we don't know. But we've been building flies here for 200 million years and have had no complaints. So I think, but that is how I do. And then, I think you have to be in possession of tremendous amount of the detail of the subject, and also know a lot of things outside of it, because sometimes there's an indication from one experiment which has no reference to this, but suddenly you realize, boy, I can explain that, and that becomes the interesting thing to do. So, the shorthand was effectively the jargon that had been developed about this corner of biology. And of course, if other people came into this, they wouldn't know what you were talking about, because there were a whole lot of words which began as lab jargon, some of which actually became part of the language. And I think that is, I think one of the things that's not realized about science, and at least, is that I think it works more like a medieval guild, in which you have, an experienced journeyman with his apprentices, and they learn from the master, so to speak. And nowadays, many people think you can learn it by taking, 49 courses and studying there, and you can do it all by some kind of teaching exercise. But just sort of hanging around and talking and discussing things is, I think, the best way of learning science. Just being there and listening to and of course that's why in the old LMB, the tea room was the open thing, and you would talk to people in other fields and learn things. And Francis was a great learner. He always wanted to learn new things, because they think they would throw light on us.
One of the reasons I was asking about this sort of special language was to lead out to ask you to describe that famous meeting in your room in King's, where only he seemed to understand what you were talking about.
Well, you see, we were talking there about an experiment. And we were talking about how does information get out of the gene. And in those days you could say there's several possibilities. One, it doesn't get out of the gene, it's used directly on the gene. Okay. Two, it gets out of the gene in this way and in that way. But we were faced with an incredible paradox at the time which defied solution. And that was the following, if I can just put it in this way. The paradox was that the little gadgets that made proteins in our cells, which are called ribosomes, were full of RNA. and I always believed that RNA was an intermediate. But the paradox was that the composition of the RNA was constant, independent of the composition of the DNA. So the DNA in bacteria could vary over a factor of 10 in the relation between the bases. Of course, being DNA, G always equals C and A equal T, so it was the ratio of GC to AT that could change from 1 in 3 to 3 to 1. And that's a paradox. And there were all kinds of solutions to it. And at that experiment, I suddenly realized that if there was a copy of the DNA which traveled to the ribosomes, because we held on to the fact that the ribosomes were the vehicles and that RNA was something, but it couldn't be tolerated, then it should have the base composition of the DNA. And I suddenly remembered that there was such a minor component described by other people in another experiment. This is the thing that was described by two people called Volkin and Ostrakhan, and many people thought that that was actually used as a precursor for the DNA. But suddenly putting those facts together with that in one blinding sort of boom, and I suddenly began to say, I said, the Volkin Ostrakhan did. Most other people didn't know what we were talking. Francis got it immediately. And I said, that carries the message. And by that evening, I had formulated the experiment to test it. And Francis had already written a paper about it that evening, but he was dissuaded from publishing it. I think we should have published it straight away. That's the concept of this, with the evidence already in the literature. And as it is, it waited for an experiment, but it was good that the experiment worked, because nobody believed it, basically. But again, I think had one said that, everybody would have poo-pooed the idea. But then it explained hundreds of facts, and actually had a physical existence. So that was very exciting. And that was... Then, there followed quite a long period till about November of that year to try to actually prove that it existed.
What was it? You mentioned there were some French people at that party.
That was Francois Jacob, and he and I finally got together it wasn't at a party, we were just having a discussion in my room. And
which room was it, do you remember?
It was E5.
Yep.
E5.
In the Gibbs building.
In Gibbs, yes. And that was a very interesting, time. And then later, Francois and I went to Caltech to do the experiment and did it in 3 weeks. But just managed only at the last week, because there's also, he describes it in his book, none of the experiments worked. They were very difficult ones, but they just kept on not working until I suddenly realized and stood up at the beach at Malibu, where we were supposed to have a bit of R&R, and said, it's the magnesium. We rushed off to the lab. But that's, it's the magnesium.
It was the magnesium.
It was the magnesium. I suddenly realized that's what's wrong.
The magnesium.
We didn't have enough magnesium. And we rushed off and we set up 3 experiments. And in one of them, the centrifuge failed. But we managed to carry, this thing had to be centrifuged for nearly 2 days. And the centrifuge we were running in had failed, and I had to carry this valuable rotor sort of through the corridor to another centrifuge where we could continue it. And that was quite a test.
So you mentioned Gibbs E5, so this might be a link to King's. When did you become a Fellow of King's?
I became a Fellow of King's in 1959. And of course it was a very interesting episode, and the reason was that Noel Lennon at the time wanted to make Francis Crick a Fellow earlier than that, and failed to get, although his name was put forward, it was not supported by the fellowship electors. Later I had the opportunity of seeing some of the letters written, and they're quite interesting. Quite interesting. So Noel wanted someone from molecular biology, and he spoke to John Kendrew, and John said, well, what about Sidney Brenner? I knew Horace Barlow very well. Horace was a fellow. And so I was put up for a competition, external competition, and although I was over age, I mean, having done so much in the time, I was not, I think that they counted my medical degrees, so they fitted in, but Noel declared me eligible. And then what happened was, I was in this competition, and then I got a letter from Churchill, which was just being founded, inviting me to become a fellow. So I thought about it at the time and said, well, I wasn't much impressed by all this college nonsense, but if I was going to have it, I wanted the real, the real authentic one. King seemed to embody that, whereas Churchill just sort of looked like an IBM studies, out there, and wouldn't be the real thing, you see. So I turned it down. Noel happened to be the chairman of the academic committee, and he phoned me up at the lab when he was told I turned the Churchill thing down. And he said, and I went to see him, and he said, well, why did you turn it down? I said, well, I'm up for the King's thing, but, because I think if I wanted the real thing, you see. So he said, but we can't guarantee it, and so on. So I said, well, if I don't get this one, I'll go without one. I mean, I'm going to need. He says, well, I have persuaded them to hold the offer open till after our election. And of course, I was elected. And I got there were two of us that got this. And so Giles Brindley got the named scholarship, the named fellowship, and I got elected under the notwithstanding statute, which I've always felt is useful to have in anything.
You've been a Fellow of King's for a very, very long time. Are there any people in King's who you were close to and who you'd like to talk about?
Well, one of the things that I did at Oxford, and that's because I got introduced to him, was a man called Dawkins.
Oh, yes.
Dawkins was a tremendous character, but of course he was a fellow of Exeter College.
This is Richard, is it?
No. This was a man who was a professor of medieval Greek in Oxford, and was well known for his relationships with Rolf, you know, Hadrian.
Oh, yes, yes.
That quest for Corfu,
Yes,
he
yes.
appears there.
Yes.
That's, of course, where Shepherd appears as well.
Really?
And Shepherd and Dawkins were great friends. So, I used to go and have tea with him in Exeter College and be fascinated by all these accounts of this. So when I came to Cambridge, I used to go quite often to have tea with Morgan Forster.
Oh, yes.
And just to, you know, just effectively talk there. And of course I had many friends there who were outside the subject of science, which is one of the great benefits of the college. You don't have to keep, you know, your social and working life is all the same, as indeed in America that is the case. So, and amongst the people I was very close to were people like Francis Haskell, Michael Jaffe. I was totally amused by Michael Jaffe.
He gave a rather frosty air to younger people sometimes.
He did, and he was also a monster so far as the servants were concerned. But you know there's a famous story about Tony Tanner and Michael.
No, tell me.
Michael Jaffe said, you must come up to my room to see my Bronzelli. And Tony thought, who the hell was Bronzelli? Bronzini? Yes. He's racking his brains to say something useful. Michael comes outside with a little elephant in bronze. That was Bronzelli. But I mean, Michael was there. And of course I very much liked A.D. Rylance. And I spent, you know, a lot of time there with a lot of people who were outside the subject, which I thought was one of the great benefits of the college.
When we first met, which was on the senior, when I was a senior research fellow, and it's the fellowship electors in 1972, 73, among the people there were Dan Mackenzie, Bernard Williams,
Yes.
Edmund Leach, were any of these? Robert Bolgar, yes, Dan Brown, Hal Dixon probably.
Yeah.
Were any of these people who...
Well, of course, Bernard was, became and was for a long time a very close friend. And I didn't put his name in because I thought you wanted to hear about the older generation. But of course, Bernard was a very important person for many reasons. And Robert Bolgar, I found, was an astonishing man, because I served with him on the electors for more than 20 years. In fact, I think that the Bolgar bottle, which is an endowment of mine, now has become the Bolgar Williams because the fund has been increased twice. I thought at the end of fellowship electors, which were rather heated, and that everybody should relax and drink a glass of wine, and this was done in memory of Robert when he died. You see, so it's called the Bolgar bottle.
Ah, I wondered how that happened.
Everybody, I mean, I still think it's being drunk now. I just gave an endowment, the value of which generated, even with the college's absurd finances, enough money to buy 2 bottles, to provide 2 bottles of a reasonable wine. So that was good, and that's nice.
What about Edmund Leach?
Edmund I knew very well before he became, while he was still a fellow of King's, and I knew him. He was one of the first people I met in Cambridge because of his interest in politics, because when I came here, I did give at some student society, I did give a lecture on Africa. And he came there, and so I got to know him, and of course his son and my son were great friends, and we knew the Leachers quite well. And I think that Edmund, and of course I knew Meyer Fortis very well. He was a South African also. Yes. And he lived next door to me in Millington Road when we first came. So I knew Meyer quite well. And anthropology has always fascinated me.
Oh, good.
Because for many years I did archaeology and paleontology as a hobby, but when it threatened to become a profession, this was in South Africa, I gave it up. But I think that and in fact one of my great interests now are to try to create a new anthropology, but I'm told we mustn't call it that because people fight about it, about the title. But I think now with all the advances in science and so on, we can create a unified subject which would be the study of, if you like, man and his works, and to try to understand our biology and our placement in the animal world, the natural world, and our emergence from that into the world of our own creation, and how our biology is not compatible with this world that we've created. So I think that's an interesting thing, and I think we could do. And since, you know, we may even have the genome of Neanderthal man pretty soon, there's enough ancient DNA lurking around that we could probably sequence it.
Fantastic. Well, returning again to your work, you came back, and at what point did you, later on you took over and founded the Institute of Molecular Biology.
The one in California.
No, the one here, or
no.
The
what
MRC.
Happened? Yes. Well, let me. I didn't found that. We were housed in the Cavendish laboratory.
Where was your room, by the way, in the Cavendish? Was it in?
It's downstairs in the basement, and we had a lab on upstairs. I've been to that room. In fact, we have been filmed in that room.
The basement in
Austin Wing.
The Austin Wing.
And we also had the little hut, which amazingly enough is still there. There have been a succession of
by the bicycle sheds.
Royce, yes, by the bicycle sheds. And in fact, I had all of that photographed because every time I've spoken to people, they say it's going to be demolished, but it still is there. It's 50 years after they've been condemned.
Yes.
However, we were there and we had become absolute masters of obtaining space. Francis would discover somebody repairing a motorcycle in some defunct room which was used for physics. He'd go to Neville Mott and say, can we have the use of this room? We were great experts in spending the weekend and converting it into a working lab. So at the end of the time, we were in 7 buildings on the site. And so, of course, prior to this, the MRC had decided that then we might have an MRC building somewhere. But we told them that wasn't a good idea, because we didn't want everybody who was in the MRC in the same building. But we formulated there that why don't they form the MRC Laboratory of Molecular Biology, as it came to be called, and that we would join up with Fred Sanger, who was in urgent need of space. Prior to that, we had entertained various possibilities, like Kendrew and Perutz would stay in the Cavendish, Francis even applied for the chair of genetics and didn't get it on the grounds he didn't know any genetics. So that's another thing people have to live down, maybe. And anyway, ultimately the MRC agreed to this proposal, and they built they were going to build out on what then was a remote site in Cambridge. Everybody wanted to be in the centre. The idea of going 2 miles out was, but eventually space was and so that building was put up, and we joined with Fred, came with us, and then we got people to come from London, Hugh Huxley, Aaron Klook to join us, and that formed the lab.
I see, but and didn't you become the...
I became the director in 1970, I think 79 officially. Prior to that I was the proleptic director, as I was called. I sometimes thought I should have been called the epileptic director. But effectively I did that till...
Who was the actual director? Or?
Max Perutz was. There wasn't a director, there was a chairman of the board.
Oh, I see.
Because there was a federation and it was very useful, but the MRC wanted to bring it all together. And so we did, of course we had lots of financial problems, but we had very serious cuts in about 1975. We lost 1/4 of our positions. It was called the Phillips cuts. It became replaced by something called the Brenner clawback. I managed to get most of them back again, so that was all right. So, and then I stopped. So when I went on, and of course it's quite a job doing all of that, and I decided that that was enough.
You said it was one of the things you regretted.
I regretted doing this because I think it was a lot of work, and not all administrative work is like that. But I decided, so when they asked me when I was 58, did I want to go on after the age of 60, because directors were supposed to retire at 60 to this and go on till you're 65, they let you go, but that's a waste of time. So I said, no, I don't want to go on after 60, I want to go back to science. So you go find a successor as quickly as possible, and he can take this and give me a small unit somewhere I'm going back to do science, which is ultimately what happened. And I then moved out of the lab into the medicine department and carried on with a small unit there, which was great fun, because it's always fun to start at the beginning again. And so we had a great time there, and then when the time came for my full retirement, I decided that retirement was like divorce. It's a question of who's leaving whom, as you may well know, but I decided I'll just stay where I am. CMRC's leaving me. And I managed to raise enough money from abroad from companies and so on to continue the lab until that just became impossible to run it. But I think I managed to find good jobs for most of my people, and I think whatever it was, almost 8 years that I spent there, was very productive in producing, a group of talented people who've now, of course, had important jobs in academic medicine and in pharmaceutical companies, and just generally I've done well.
Can we go back to your work on the elegant nematode?
Word. Yes.
How did you get? You sometimes said that even more important than the picking the right question was picking the right subject.
Yeah, I think picking the right thing to work on. Well, you see, that I'd had the experience of bacteriophage. Bacteriophage was a superb example of, by reducing the complexities while preserving the essence of the problem, which is how does DNA replicate, allowed one to tackle it directly in what's now come called a model system. And so, as long as that preserved the central problem, then it was the correct sort of model system. It had to satisfy a huge number of conditions. One, they were easy to keep, easy to work with in the lab, because to introduce something like that against the whole background of biology with, established things, but I decided, well, I didn't care, this is what I wanted to do, and I was jolly well going to do it, because I had formulated what was the essential question, and it was the question, it's the same question that we're always talking about, which is, roughly speaking, if you had the wiring diagram of anything, could you actually use it to compute the behavior? In other words, if I knew the pattern of the nervous system, that's all I would have to know, and then I could work out how it works. By the way, we did do the wiring diagram with C. elegans. It took us 25 years to do. It was a group of very talented people and hard-working people. I think maybe the second problem is now, because there are lots of people in the nematode field, the second problem will probably be solved in the next 5 years because of just the nature of the organism. So
the second problem being...
Second problem, how does it work?
Yeah.
All right? How does it work? Can you actually take it and run it in the real world? Now, but it made me think very carefully just about the methodology of all of this. And you see, one very striking thing happened is one day Jonathan Hodgkin, who was Alan Hodgkin's son, my graduate student, now professor at Oxford of genetics, came in and said, oh, I've just done a nice simulation of the worm's behavior. And he showed me a screen, a computer screen, and there were all these things wriggling around, you see. And I said, that's very nice. And I said, can I see the program? So they produced the program, and I said, this isn't a proper simulation. This is a description of the behavior. Of course, all I can see is cos theta, sine theta. You're just generating waves. What I want to see is muscles, neurons, stimulation. I want to see how the meat is working. So, they can now distinguish then between a simulation of the behavior and an imitation of the behavior. That's an imitation. And I think that to me immediately solved all the problems about that everybody's argued about the Turing test for an intelligent machine. Because the way it's posed is there's no machine that's programmed with real sort of simulated neurons and so on. There's just a machine imitating an individual, answering the questions. So it doesn't solve the Turing test, because you have to, in other words, you have to open the box to see if it's a proper simulation. So I would want to, so to speak, if I had a machine in one room and a person in the other room, at the end of the day, I don't care if they are the same. I want to see who bleeds when I stick a knife into them, for example. But they should be, if it's to be scientifically valuable, it needs to actually use the machine language of the object being simulated. And that's become very clear now in doing all of this. That's what we got to get down to do. But I mean, I think there are dozens of very talented young people in the field, and I'm sure that problem now will be solved.
But having chosen your nematode, what your main work was trying to get the wiring?
No, I think we decided we had two problems. One was, you see, everybody was saying, well, there's genes and behavior, right? And of course many people were saying, well, how do you go from genes to behavior, you see? And of course there were lots of discussions about this, but effectively that problem is very ill posed, okay, because you say, what do genes do? So genes build nervous systems. I shouldn't actually say they do, but the information in them is used to build a brain, a nervous system, which then performs the behavior. In other words, there is a construction phase there, and unless you understand that, you cannot go from one to the other, because the mapping is through this construction. So you must understand that. So therefore, when we wanted to do this, we wanted something which we could determine the structure of the nervous system, that it should be a small nervous system, which means it should be finite. That is, we could say we've come to the end of description, and that we can then look at, as we thought then, we would make mutations and see how it altered the behavior. And then we would hope to be able to see what changes in the nervous system these mutations produced. And then we would be able to map those onto the altered behavior. Now, that program has been partly carried out, but effectively it involved doing the anatomy, it involved doing the full embryology, and the big advantage of these nematodes is they had from the literature stereotyped nervous system, constant number of cells, and the same, it was thought, in every single nematode of the same genetic composition. So that meant we could ask ourselves, under what conditions, how accurately do you build a nervous system with the same genetic program? In other words, are there mistakes made in it? Is there flexibility? Is there noise? And so all of those questions could be decided with this little beast. It was easy to keep in the lab. Francis used to say, Sidney likes nematodes because they wriggle. You can actually watch them. But to me, what was really important was, with a few simple tools, a good microscope and some toothpicks and so on, anybody could start to do what became sophisticated genetics of metazoan organisms themselves in a lab by themselves with the hot paraphernalia of mouse houses and all the other stuff. So it was this, and it has caught on, as you know.
Is this the work for which you got the Nobel Prize?
Well, the Nobel Prize, it is the work in part. Nobel Prize gives the prize for discovery. So I discovered something which had been discovered a long time ago, but it doesn't matter. Yeah.
How do you mean it had been discovered a long time?
Well, I mean, their genes had something to do with organ formation, but it was the structure of it, which I think was very nice, in that they included John Salston and somebody who'd made really novel discoveries about cell death using the nematode. You see, actually, if you said you wanted to give a prize for cell death, which was given, then you work out that effectively John Salston was the one who discovered there was normal cell death. And of course neither of them could have done the work unless it had already been established as a genetic organism in a lab. So
And the third person was...
Bob Horvitz, yeah, American. And so I think this was good. It's not... so that was it was good.
I was very struck by all sorts of your aphorisms about how research is done, one of which was the don't worry hypothesis. Can you explain what the don't worry hypothesis is?
The don't worry hypothesis is the following. There are classic examples of it. You simply have to find a plausible biochemical basis for it, material basis for this. And then it says, don't worry, there is at least one thing which could be the basis of it. Let me give you the most famous don't worry hypothesis, okay? People worry because the DNA strands are wound around each other. Therefore, they said it's impossible that this could be copied because the old strands would have to be unwound. So we said, don't worry, there'll be an enzyme which unwinds the DNA. Boy, there sure is about 24 of them that unwind DNA. So that's a good don't worry hypothesis. Then you can get on with the core. But if you worry and say, oh, well, this is going to form a barrier to further progress and it must be wrong. So the don't worry hypothesis is very useful.
So it's really making a guess and not worrying that it's absolutely...
No, it's making a guess which says you can take that away from your present problems and try and solve the others. So don't sit and worry about this problem. See, replication, if you worried about that, you'd say, oh, well, that must mean the whole of the DNA structure is wrong, because it'll never work. Right? So all you have to do is provide one plausible hypothesis. I did this again for immunological variation. In fact, we wrote it up. It was, somebody had written a paper to say it is impossible, you can just mutate parts of the gene. All right, so therefore that must be wrong. So all we did is provided a plausible mechanism based on good evidence in neighbouring fields that it could be like this. But what we've shown is it's not impossible. It may not be true, but it's not impossible, you see. Therefore, you needn't worry about that side of the thing and get on with the rest of it.
I would sort of rephrase it in a way as a mental pending tray.
Yeah.
You put things in the pending tray, and it reminds me of, I often ask my students, what was the greatest invention of the 20th century? And they puzzled for a while, and I said, well, according to Bertrand Russell, it was the suspended judgment. Saying we don't know yet, but let's just leave it.
Well, you better not have a pending tray like me, because I always hope the P falls off so it becomes the ending train.
The other thing, another of the many striking things that I liked about your book was the virtue of ignorance, which we touched on before. It can't have escaped you, as it hasn't me, that many of the greatest breakthroughs have been done by people on the edges of disciplines. And I'd always assumed that this is because they would ask the very simple questions, which professionals had become forgotten, the questions within their fields. Also, they were obstructed by the sort of thing you mentioned, which was that they knew too many of the objections to every argument. I've been taught by many people who can destroy their own arguments before they're formed even.
Yeah.
But is that what you meant by...
Yes, I think ignorance is the following, you see. If you don't know all the sort of things, then you never try anything. At least in our experiment, I mean, and so if you did, for example, there was an experiment done by a postdoc of mine, and my judgment was, because I know a lot, that he was unlikely to get it to work. But I said, have a try. So he went into the cold room, he took 2 bottles. We did the experiment and it worked. I then went back later and calculated what was the chances of him picking up those 2 bottles. The chances were about 1 in a million that he would get those 2 bottles. Because there were 1,000 bottles there and he'd have to get the right 2, so it's about 1,000 square. And of course, he just did it. And I asked him, how did you pick those? He says, they were in the front. But that's why, but he didn't know how difficult it was. But I held myself back. So he was ignorant. He thought, well, he'll just do this and something will happen. But of course, the grin on his face when it actually worked. But later we found that there was a sophisticated, more interesting way of doing it, ab initio, without having to take chances. But we've never come to that without going through this. So one always, as you rightly say, one always knows what's wrong. One can always produce arguments to demolish one's thing, but it just means that one should persevere. And of course, if you are wrong, you'll find out sooner or later. But I notice that as soon as people become too knowledgeable in a given field, they are always concerned about the difficulties of doing it. And you know, this has been tried before, it hasn't worked, no one has done this. But I think that's why I think ignoring all of that is basically a good thing, because I think that's the only way you can innovate.
There were just 2 more things I wanted to ask you about. One was, quite recently, you've not only been working quite a lot in America, but also in Singapore.
Yeah.
What is your work in Singapore?
Well, I went out there in 1984. They wanted some advice on biotechnology, and I told them you better set up a graduate department in cell and molecular biology, because you won't do biotechnology until you've produced a surplus of PhDs in that subject. And I've been involved with them, and then of course very recently, starting in 1999, there was a huge surge forward in this, and so I've been heavily involved in that, in building up a gigantic operation there. And of course, one of the amusing things about there is they built a huge set of labs there. It's called Biopolis. But when I first suggested that name, and of course in the Singapore accent it was pronounced Biopolis, which sounds like the police to gardeners. Actually, Singapore accent is very difficult because you cannot distinguish between an air ticket and etiquette.
Right.
So I have just retired as chairman of the council, and I've now, as in Singapore, you know, you get promoted to be senior X, then mentor X. Finally, you become dementor X.
And you've been given various awards by the Singapore government for this work. And the last question is, which you may have sort of answered, but if you were giving advice to yourself as a young scientist in your 20s, starting again or starting now, what would you tell such a person as to how to proceed and to do interesting and valuable work?
Well, I think it's very difficult to do it now because science has become a highly structured profession with many more people in it, many more institutions involved. Of course, all scientists are now, of course, involved in getting funding. So I think you would have great difficulties in trying to do something as a young person off your own bat, because you need so much, you know, just actual material and financial support. And so I think that's difficult. But what I would choose, I would tell such a young person, is to go to a lab, not because it's a famous lab, but where there's a good mentor, somebody that you can really get the benefit of, that will help you. In America it's rather difficult because a graduate student is the source of semi-skilled labour, some say unskilled labour. So I think that's very difficult to do there. But I know what I did was simply go, if I got interested in the subject, I simply went and got a book out of the library and started to read. And there's quite a lot you can do, which I do now, which I think is fascinating, which I'm very deeply concerned with a very interesting question about how we can reconstruct the past. And in fact, a few months ago, I organized a meeting on this for the Gulbenkian Foundation. And we started with the origin of the universe. And of course, I now think that we have available for free all the sequences of literally hundreds of organisms now, genetic sequences. And so I think it's a big challenge. That's what I do now, is could we reconstruct the past? You know, could I actually try to work out what happened before the Cambrian? Because I can now tell quite easily where we started 500 million years ago, vertebrates like us. But could we go a little bit earlier than that, to the origins of complex metazoans? And I think that to me is the fascinating thing that I think you can do, because computers are cheap, they are available. And I think there are a lot of subjects that you can do simply by going to the book to do this. But in my subject, which is such an experimental subject, what has now beginning to decline in it is the art of the individual solving problems. You see? I mean, one thing I've always said, and that is people have tried to divide science into pure and applied. or as some wit once said, the difference between the applied and the applicable is the same as between the despised and the despicable. But that is neither here nor there. But of course, what science is a way of solving problems. It doesn't matter whether it's applied, and it doesn't matter what it does. So we should be judged on the quality of the problems we choose and the quality of the solutions we find for them. And that is something that I think really works. As I keep on telling people, science works. It's the best way we know of solving problems. I mean, magic doesn't work. Religion is very unreliable, but science works. And so I think that's what you have to do. What is the problem you're trying to solve? And I think for young people, they should ask themselves that question. What are they burning to understand the basis? I mean, till his dying hour, Francis was interested in solving the problem of consciousness, which I don't think is a soluble problem personally. I think it's one of those great problems that'll just disappear. And 50 years' time, people will say, what the hell were they talking about? What were they worrying about? However, so I think for a young person, find a good problem and work on it, and then try, of course, you have to gain entry into the whole apparatus of science, which is difficult. Because I've always felt maybe what we should first start when we start a new lab or a new institute is a department of bank robbery. If we could master that, we wouldn't have to worry about grants or all of the other things.